Okay, so they are recording. Oh, and I have to make you guys co-hosts. One last thing here.
Bill, we just ask you to next slide.
Sure, we can do that. That's not a problem. Tim, I don't have your stuff, so I'll just make you co-host. You should be able to share.
Yeah, okay. It's
Actually, you know what? I'll just do mine because I can move my.
okay.
Yeah, I mean, whatever is easier.
How's that for a picture?
Ooh, nice. If I didn't know any better, I think that was from Colorado.
Yeah, it's letting me share, so that's good.
Okay. Deal. I got to share my stuff. Okay. Oh boy. And I want. Stop share. Okay, I think we're good here. I got 18 people coming in, so I'm gonna go ahead and start letting people in. Well, good evening, everybody. Evening for some of us anyway. From where I come from, it's 6 o'clock here in the Chicago area. For those who don't know me, my name is Phil Ash. I'm a board member of GRIN2B Foundation, and welcome to this webinar. The plan is we're gonna go for about an hour and 15 minutes. And I just have a couple of things to share with you all before I turn it over to our guest participants this evening. So I'm going to share my screen. We are recording this, so kind of keep that in mind. May want to just kind of keep keep yourselves muted unless you do have a question. I'm going to ask that we hold questions until the end, just in the interest of efficiency. Let me go ahead and share my screen here. Hopefully everybody can see that. Let's see. Okay, so this evening we will be discussing sort of different GRIN disorders and this idea of gain of function versus loss of function. And so that'll be sort of the main topic of conversation for this evening. So again, thank you everybody for joining. And then lastly, just one last thing before I turn it over to our guest this evening. Those of us in the GRIN2B community, we have a community needs assessment survey, and we're asking all of those in the GRIN2B community to take this. It's part of our sort of strategic plan that we're undergoing, and so your voice is very important into how we sort of shape what our plan will be in the coming years. The QR code is here. In just a moment, I'm going to copy the link to that survey, and I'll put it in the chat so that you guys can get it. So if you want to take a moment to scan that, or I'll put it here in the chat here in just a second. And then always, yeah, thanks, Tim. You guys can obviously, any questions you might have, you can put in the chat as well. I did put the link to the survey into the chat as well, so that's been done. And I got to admit a few other people. Okay, so at this time, I'm going to turn things over to our guest this evening. So two of our researchers are here to present some information on gain of function versus loss of function. I'd like to turn it over first to Dr. Steve Traynelis, coming to us from somewhere in Georgia, now Emory University. And so he's got a slide presentation that he'll share with us, and then he'll be followed by Dr. Tim Benke, who is coming to us from Colorado. And without further ado, I'll turn things over to Steve, but just again, real quick, if you have questions, please put them into the chat. And kind of in the interest of efficiency's sake, though, what I would like to do is have Steve present, followed by Tim, and then we'll take questions at the end. We all good to go?
I think so. Is that Phil, does that look good on the screen?
Yeah, sure does. I can see it great.
All right. Good to be here, everybody. And my name is Steve Traynelis, as Phil had mentioned, in Department of Pharmacology, Chemical Biology in Atlanta at Emory. And I've got on this slide many sources of funding, as well as on the bottom are my competing interests, just for clarity. Okay, I'm going to tell you about this idea of gain of loss of function, but to do that, we sort of have to step back a little bit and talk in general about the NMDA receptor. And I think everybody here has been digging in and learning about that. And so I apologize if some of this is redundant, but just to synchronize our thinking, we'll sort of go back to the beginning. The NMDA receptor is a member of a large family of glutamate receptor subtypes known as delta, AMPA, kainate, and NMDA. And the NMDA receptors are encoded primarily by 5 genes: GRIN1, GRIN2A, GRIN2B, which of course everyone here is familiar with, GRIN2C and GRIN2D. There's a couple of GRIN3s we're not going to talk about anymore today. And these receptors are present in all neurons in the brain and in the spinal cord, and very likely all neurons in the peripheral nervous system, myenteric plexus around your GI and things. And they're important for a large number of processes: elementary communication between neurons, which we call excitatory synaptic transmission, and then higher level things everybody here is familiar with: learning, memory, movement, as well as the development of the brain from the beginning all the way through life. What I've got here is a slide that summarizes how I look at these and how I think about their function. And so up on the upper left here is just a stylized cartoon of one of these connections between neurons, one of these synapses, and below is actually an electron micrograph of a section of rodent tissue showing what they really look like. This is a terminal at the end of a long axon from a neuron. These gold balls are meant to represent glutamate-filled vesicles that are released when this neuron is depolarized and decides to talk to its neighbor. And that glutamate enters a very narrow cleft, about 30 nanometers. They sort of have a cloud of yellow glutamate. If one could go in and measure the glutamate, the neurotransmitter, in that space, one would see that it very rapidly rises to what we consider a high level. We call it 1 millimolar. But within a few milliseconds, it's all removed by a variety of processes. So you just get a little tick of neurotransmitter. And on the other side of this terminal is a postsynaptic connection. We call it the postsynaptic density. And it is embedded with receptors, glutamate receptors, and a lot of other signaling proteins that control how this neuron is going to respond to that. And if we just think about these two channels, these two ion channels, these two receptors, NMDA and its family member, the AMPA receptor, I've shown what they might do if one could look at the electrical current they mediate. And these channels are, these receptors, when they bind to the neurotransmitter glutamate, basically open a hole in the membrane, ions move through, and it's like injecting current into the downstream neuron that's being spoken to. If one could stick an electrode in and measure that at high temporal resolution, as microsecond resolution, what one would see is that little tick of glutamate initially activates very fast, activating and very rapidly inactivating or shutting off AMPA receptors. And those mediate most of the communication in the brain. That's what's allowing me to talk, to move, or you guys to think. That's the standard mode of neuron to neuron excitatory communication. If I then looked later after this AMPA receptor, normally I would see nothing. But if I experimentally removed all the extracellular magnesium and divalent ion that's present, or I depolarized that cell, I would uncover a large, long current mediated by NMDA receptors. And if you look at the area I've colored light blue, it's a lot more than the area of this gray AMPA current. And it has the added property that it carries a different ion than the AMPAs usually do, calcium. And that allows NMDA receptor activation to be coupled to all kinds of intracellular processes. It's important in changing the synapses, learning, and memory. And so the NMDA receptors are special. They mediate calcium signaling, and they respond to that depolarization I meant, and the release of glutamate. And so, you may have seen in reading for background, sometimes people call them coincidence detectors. There are, as I mentioned, there's 5 genes, and these form tetrameric assemblies, 2 GRIN1, 2 GRIN2s. And because we have 4 GRIN2s, we can make 10 receptors. And I've just shown how they're paired up. I've put a blue asterisk over the ones that I think this audience is mostly interested in. A red asterisk with these blue 2Bs, and you can see a receptor might have 2 copies of 2B, or it might have 1 copy of 2A, 1 copy of 2B, and so on. And so these would be the receptors that would be affected in any sort of genetic change that altered the GRIN2B gene. I've just put this picture up just to give you a little bit more feel for the receptor. It's not just 4 little cylinders stuck together. This is a crystallographic X-ray structure of most of the NMDA receptor. Part of it's missing. It's a machine that's built of about 5,000 parts. These little helical coils are rods, sometimes rigid, but they can have hinges hidden in them. There are loops and sheets, and proteins like this have the added feature that a lot of physical material we play with, here, you imagine Lego erector set, you just bolt stuff together and it sticks. Well, proteins can have slippery parts, greasy parts, parts that repel each other, parts that attract each other. So there's 5,000 parts interact in a much more complicated way than a machine we would build with 5,000 metal parts. And that's the main point of this slide. It's a complicated machine. And I like to use this analogy of a car. A car motor, a car engine, it's about 5,000 parts. And when we think of the genetic variation where you might have a error in the actual copying of the GRIN2B gene, you might have one of those parts no longer be correct. And so if we just think of a car, if we had 5,000 parts of a car, we could ask the question, what happens if at the factory we had a part swap by accident? And I think I got up here the distributor cap pulled up. One of 3 things could happen. If you just popped in one part and then you assembled your car and you ran it, either that part might make the car work better, the car might go faster, it might be more efficient. There are a lot of ways one could come up with a reason why you might think it would work better. Could also make a car work worse or not at all. A car might not start, it might run rough, you might not be able to go over 10 miles an hour, might burn all your gas in half the time, and it might do nothing. And so those 3 options, one could use the words gain of function when that part swap makes the car work better, loss of function when it makes it work worse, or tolerant when there's no effect. And I like this analogy because we can directly convert it to a missense variant in a GRIN2B gene, where rather than the distributor being swapped out at the factory, 1 amino acid residue is swapped out at the factory. And I've just colored here. Here's that structure of GRIN2B. I've colored in blue the regions that these variants that can cause problems usually occur. And if we just had a change right here in red, one might imagine that could either make the receptor work better, it could make the receptor work worse, or the receptor might be tolerant to a change in that position. A key concept to try to emphasize is these are really complicated machines. And there's a lot of different ways you could say a change makes them work better. Just like a car, it could run faster, it could run more efficient, the air conditioner could work better. There are lots of ways you might say, I improve the function of that car, but all with different properties. So what we do in my lab is a lot of electrical recording, and we do a lot of cellular and molecular analysis, and we like to measure lots of things that receptor does. And so in trying to understand these variants and understand that part swap illustration, we make 6 measurements of this protein because it does a lot of different things. It's activated by glutamate, so we measure how sensitive it is to that. It needs glycine binding, another amino acid, not a neurotransmitter, but something that's in the background and also dynamically changes in the brain. We measure its sensitivity to that. I mentioned this extracellular divalent ion that's present in all cells and outside the cells, magnesium, that blocks the NMDA receptor and gives it a really special property of being a coincidence detector. We measure that. We measure the time course. Over here, there's an arrow. I don't know if you can see my mouse, but deactivation time course, how fast the glutamate comes off. That defines that the length of that current I showed you a few slides ago. We measure how efficient that hole I talked about is in terms of being opened. And then we also try to determine, did these part changes alter how many of these receptors actually get built and go to the surface? And sometimes they do. There are more things we could measure, but we sort of assess those as 6 of the really key properties that we can measure. Now, I've shown over here on the right a stylized synaptic current, and in green might be a normal NMDA current you might measure in any sort of neuron. And you might then have that part swap that makes the receptor gain of function work a lot better. And you can see now the red current stays open for much longer. And all the area under this curve in green is the current injected. And so you can see there's a lot more area with red. And so this receptor is just passing a lot more current. And we can quantify that if we just were to calculate the areas under those 2 curves. And it's not, if you're not mathematically inclined or you don't love math, it's a lot easier than it might sound. All we need to know really is the height, is the height of that signal, of the variant relative to normal. And then we need to know the time course, how fast this goes away compared to normal. And we can calculate that from these 6 properties. So we make those measurements for as many variants as we can. There's going to be 1,000. I think we're up to 800 or 900 that we know about. And we tried to come up with a way to boil them all down and decide when you measure all those, how are we going to decide if the car ran faster or the car ran better or the car ran worse? And so what we settled on is this discrete analysis of all 6 parameters. And we classified the changes based on statistics as having, high confidence of being important, moderate confidence, or no effect. And then we came up, and this boils down a lot of work from Tim, Johannes Lemke, others as well over many years. And we sort of boiled down all of these analysis into a set of rules. And when we find any one of the 6 parameters changes with high confidence and nobody else changes, that's a likely loss or gain of function depending on the direction. If you get a couple of those parameters change with moderate confidence, you can be pretty sure that's going to either be loss or function as long as there's not conflicts. And I'll show you that in a minute. If nobody changes, it's no detectable effect. And we don't say benign because the receptor does other things we haven't measured that could have been affected. And then sometimes, as you'll see in the next slide or two, you get one property go up, one property go down, and you have this conflict. And we have some tiebreakers for those conflicts. And sometimes you just cannot, there's no clear evidence statistically, and we call it indeterminate. And that word was carefully selected. It's not that it's, complex, they're all complex. It's that at this point in time, we can't figure it out. It doesn't mean it doesn't do anything. It's just doing something that doesn't easily fit into the simple categories. And then we have one more category where we basically can't make any measurements. The protein is made, it goes to the surface of the cell. We put our electrodes in, there's absolutely no current, and we just say, well, that's clearly loss of function. The channel is simply not working for whatever reason. And so we have these rules that we've set up. These have been published recently in Human Molecular Genetics. And we've been sort of going back and applying these rules this year to all the work we had been doing since we started this about 10 years ago. Here's an example of some of those numbers. I don't expect you to look at all this, just kind of look at the colors. These would be missense variants. If we just look at this first one, isoleucine 150 to valine, a change, you can see we've categorized glutamate potency, glycine potency, magnesium potency. This is basically efficiency of the channel to open, speed with which it shuts, and how much gets to the surface. And here's a great example of conflict. Looks like glutamate's a lot more efficient as a gain of function, but the channel doesn't open as well. And so, we would score this as conflict, where the next one is pretty clear. You only have things that decrease function. The glutamate is not as effective, and the channel doesn't go to the surface. And so, we can add these up and look across our rules and come up with these classifications of indeterminate, likely, loss of function, possible gain of function. Now, I mentioned we have a tiebreaker, and the tiebreaker took a long time to think through and develop, and I think it's a really necessary part. And I'll just take a minute to explain it, and then I'll kind of show you how this applies to that synaptic current and turn it over to Tim. Sometimes we'll see somebody like this tryptophan 607 serine, where we have a couple of losses, a high, and so we're in conflict, not really sure which direction it's going to go. A lot of things look like they're worse, but one of them looks like it's a heck of a lot better. And so because with our discrete analysis, it doesn't fall into the rules, what we can do is go back to that math that I mentioned where we calculate the charge transfer, the area under the curve. And so one of our tiebreakers is if we use all of these parameters to figure out how much the current changed, then we can make a call that, the net effect is gain or the net effect is loss. And so when we have a conflict, but our synaptic charge transfer is really clear, that promotes the conflict to a categorization. And we have another tiebreaker called non-synaptic charge transfer I won't go into unless it comes up in the questions. So that's a quick description with, I think, a simplistic, example of a car, and then a little bit about what we measure, and then how we take those numbers and try to come up with a system, because it's a complicated machine and we have to measure all these things, of calling gain or loss. Now, I have a couple of caveats, and then I'm going to go back to that synaptic current. I'm going to show you how I imagine gain and loss might look. So caveats, there's a lot of things this receptor does. We just can't measure it all. And some of them are a little bit rare, and so we left them out. But receptors desensitize with time. They work less and less effectively. And these missense variants can change that. And we know how to measure that. But so far, we've only seen that happen in a couple of our variants. And so when we think about doing 1,000 variants and our time and resources, we sort of imagine that's not a high priority, but it's a caveat. It could be something we missed. Ditto with extracellular modulation by ions like zinc, very important and actually very interesting. We used to work a lot on zinc. I've ignored a large piece of the receptor that sits inside the membrane and talks to all kinds of other proteins. None of our assays really speak to changes in how that is embedded in the synapse or how it might mediate downstream changes for memory. And then our expression systems are just non-neuronal cells that express, one given receptor. They don't really recapitulate what's going on in neurons, either in the context of human development with, trillions of neurons talking to each other, or the context of neurons having their own set of genes that are expressed compared to the cells we're studying. And then last of all, we picked 6 things, but it's really complicated, and again, you can think of a car, your car stops working. There's a lot of reasons why that could happen, more than 6, but there's 6 highly likely ones. And when those don't really show an effect and the car's not working, there's still something else wrong. Well, these receptors, many of them, we can't quite figure out what's going on, but that doesn't mean, there hasn't been a change in their properties that we just haven't assessed. So let's just end with that initial slide here. And if you just focus on this lower part, this blue current, remember this is how a trace as a function of time when one neuron talks to the other and releases glutamate, and these NMDA receptors are activated, and you see a slow rise and a slow decay, and that's current. Those are positive charges, cations, sodium and calcium flowing into the neuron downstream and depolarizing it. If you were to have a gain of function, you might expect something like that. That current might just get a lot bigger. and there's an ongoing trial for a GRIN2B selective inhibitor, Registry. You might imagine if one were to take this circuit, just this, single connection, and ask, well, what does Registry to do a gain of function? You might imagine it pushing you back in the direction to where you started. Maybe not perfectly, but attenuating that exaggeration. And then if we just go back to that baseline, what would a loss of function look like? Well, a loss of function just might be a lot smaller. And now, and I like this slide because it sort of emphasizes why we want to know this bit of information. If that's already small, you probably don't want to put an inhibitor on that connection, because you're not going to make it any bigger and get it closer to normal. You're simply going to push it further away and make it even smaller. So we would predict an inhibitor has a chance of reducing that signal further and potentially exacerbating some of the problems. So just to summarize, the variants are these missense variants encoded by the GRIN2B gene. It's like a single part swap of some complicated machine. They can have many effects on receptor properties. We need to measure multiple properties to figure things out. These variants can change any or all of those properties we measure, and so we have to create a way to make sense of that. And we've done that with these multiple assays and this sort of discrete gain or loss of function analysis that I described. The functional evaluation of these variants, we consider it central. It allows stratification of patients for clinical trials, the example with Registry that I just gave you. And in fact, down the road, I'm optimistic that as we learn more and more about this and the patient registries grow and we catalog all the variants in terms of their function, I'm imagining a time where clinical symptoms will not just be correlated with gain or loss, but we'll ask what happens in patients that have just this subset of changes or another subset of changes, and we'll further stratify the patient population, understand what works for them, not just with treatments, but in lifestyle. I'll stop there. We've done about, we're just on the edge of bringing about 200 of these variants fully up to this form of analysis. And I just have my slide of collaborators up. So let me stop sharing, Tim, and pass it over to you.
Yeah, thanks, Steve. So thank you, everybody, those that have come in late. We're again going to ask, if you have questions, go ahead and put them in the chat. I'm going to ask to withhold those until we get through Tim's presentation. So now that we hopefully have a little bit of a better understanding about gain and loss, Dr. Benke is going to explain sort of why gathering that information is so important and what our medical research team intends to do with that information. So without further ado, Dr. Benke.
Thanks, Steve. I thought that was really clear. Thanks everybody for inviting us to chat to you today, and I hope this is useful for you. So I hope you can see my screen now. There's not. Okay, good. So we're going to talk about what to do with this information once you might have a report about gain or loss. So let's talk about that. So here are my disclosures. Nothing really hugely impactful on what we're going to talk about. So, as Steve was mentioning there at the end, the overall hypothesis is that whatever your change is, that's going to change your function a certain way, and that's going to influence what your symptoms are and also potentially what the treatments are. And this is the long list of things that I think everybody wants to know about. If I have a certain change in my GRIN, what do things look like, and how can we as clinicians work with that information to have a personalized approach. So I think the question that everybody's asking is, where is my functional analysis? And so the passport to getting that is to enroll in our registries, and I'll tell you more about that. But then once you are enrolled in the registry and you've completed your questionnaires, I'll tell you the pathway, but basically your physician can email me and we can talk about what that functional analysis shows. The reason why we're not providing it directly to you, because then implies a sort of different relationship than the one that you signed up for when you joined the registry, and that's a research relationship. And our goal is to eventually put all of these in the GRIN portal, and we'll tell you what that looks like if you haven't been there already. So the gain of function versus loss of function has been reviewed by Steve, but at the moment, this is not clinically actionable. You know, I think as we move forward in time, we would like for it to be clinically actionable, but we're not there yet. But it's necessary to get there through clinical trial research and natural history studies. So an example is in a natural history study, the gain of functions might have different symptoms at different ages compared to loss of functions. And another example that Steve illustrated is that future clinical trials may only include gain of functions and exclude loss of functions, or vice versa. And that's specifically because of that reason. An example that Steve showed is that the drug is likely to be specific for one versus the other, and the wrong drug may make symptoms worse. And that's the last thing we want to do. So this is what the GRIN portal looks like, and you can find it if you just Google GRIN portal, it'll come up, and there's some, you know, basically different tags here that can get you into the portal and give you different information. But usually folks will go to click on the variant analysis. That's where eventually the functional reports are going to reside. At the moment, there are some functional reports, but they're not up to the new standard that Steve has created, that is going to be what we think is much more clear than what's currently available. This is what happens when you hit the family organization pages, which includes you know, our favorite people here. So, and then this is what you do if you want to participate in one of the registries. And basically we're segregated into the Americas and Australia's, participate through our registry here, and there are the links over here, whereas everybody over in Europe and Asia goes through the registry sponsored by Johannes Lemke. So always about the registry, why haven't you enrolled and why haven't you updated your forms yet? But basically that link for families in the Americas is through GRIN research at cuanschutz.edu. And once you do that, you're then asked to email full copies of your genetic report. So that's all pages. And then Dr. Park and myself, we review whether or not you're eligible for enrollment. Our study coordinator then will email you links to an online consent form. If you have any questions about that consent form, you should be asking those, and then Dr. Park and myself will look at those. The study coordinator verifies that you signed the consent, and then they send you links to the GRIN questionnaire. That's what TGQ stands for. Once you complete that, then we notify Emory to proceed with the functional analysis. I only have one study coordinator. She's on the job Tuesdays and Thursdays. And at this point, sometimes this whole process just to get enrolled may take you 3 to 4 weeks, but we do the best we can. And Steve can comment more perhaps in the questions about how long functional analysis takes. But again, we have limited resources. This is not like a clinical test. It's a research test. And as you can see from the many moving parts in that 5,000 part engine that Steve's interrogating, it takes a while to get there. So this is where we are with completed questionnaires. And as always, you guys lead the pack with how many folks enrolled in completed questionnaires that we have. And what's important really with this is that we've been waiting to get to basically this particular threshold where we have more than 50 in our different groups, because we just think that this is going to be a good number to start to make comparisons. And so this is a good place to be and what we're working on at the moment. So this is that link to the portal. And again, these are those links to enroll in the registry. Again, for Europe, Asia, Africa, this is Dr. Lemke. For the Americas and Australia, this is us. There's also a partner registry at Simon's Foundation through Jennifer Bain. And basically, we're asking different questions. It is nice if you're able to do both, because we do have this, we are talking to each other and combining data. So the next thing to chat about is something that's sometimes called N of 1 trials. And this is basically where you are your own experiment, is basically what that means. And it's an individualized approach and outcomes for each person, and there's often not comparisons amongst bigger groups. But what the challenge is it's always difficult to know, well, how are you going to measure success? How are you going to know if something's working? And also importantly, how are you going to know if something is actually causing you problems? And really, this is something that we all struggle with in the clinical trial space. It's not really just about knowing that if group A versus group B is statistically different between the two groups, but was that change between the two groups actually meaningful to the people that were treated? And this is something that is typically very personal. And it's not always clear that prior safety is established. It might be established with a particular drug that's already FDA approved, but it may not be approved in a certain population or known to be safe in that population. And so it's not clear whether things are safe and how we actually assess safety. And the other big question is if a symptom is worsening or improving, how do you know if this would have happened anyway? In other words, it's part of the natural course of having a GRIN2B disorder. So was it the medicine, or was this going to happen anyway? And so these are really difficult questions to answer when you're doing individualized N of 1 trials. An important thing to know is that the placebo effect is going to happen. We know that this happens in blinded trials in very severe disorders. An example that I just put up here is with Rett syndrome. But basically, here was the outcome measurement on this axis here. Here's during days of the study. Green is the folks that got drug, and the dotted line is folks that got the placebo. Well, a downward deflection is actually improvement. And it's clear that those that were taking placebo felt like that they were getting better in the trial. So even when the study is blinded and you don't know what you're getting, a placebo effect is something that happens. So if someone is to contemplate an individual trial, these really depend on the prior knowledge of functional status, whether or not you're gain of function or loss of function. We know from small studies that for loss of function, that supplementation with L-serine may demonstrate improvements in behavior, development, EEG, and seizures. And it's really relatively small numbers. And of course, this is open label, and the outcome measures were not standardized across the different individuals. But basically, in general, there was a study for 9 people published by Cray, and in general, 8 of them got better. One person got worse, but it turned out that they had a gain of function. So as Steve was suggesting, you want to make sure that the right person is getting the right drug. There's another study from the Soto group, and again, it was only 1 patient. For gain of function changes, such drugs can include things like memantine, dextromethorphan, magnesium, and ketamine. And basically what's been found is improvements in medically refractory epilepsy, and especially status epilepticus. And again, these are all relatively small studies. One patient here, 4 folks here, 1 here, another 1 here, and 2 here. So these are relatively small studies. And basically what they're telling us is, hey, there is probably a signal here, but we really need to sort it out so that we can quantify it. And then when you come to your doctor, such as myself or somebody else, we can talk about that. We can say, well, here's the sort of change you can expect. Are we expecting a single, or is this just a home, or is it going to be a home run? You need to know that. That's information that you need to know in order to make informed decisions about taking care of your loved one. So these are my opinions, and I think these are helpful to understand safety and support going forward with standard clinical trials. And larger trials are needed to understand, who responds and how much, and especially what is safe. And I think that for these small trials, that these should be for extreme situations where somebody is really sick, they're in the ICU, they're having to go to the hospital over and over again, having a lot of seizures, and when the functional status is known. And I often, in fact, I was talking to clinicians today about somebody who's really sick in such a situation, and they can get in touch and we can have a conversation. And also what sometimes happens is families email me, and then we figure out how to get in touch with your clinician, and we go from there. So until a real trial comes along, here are some issues and suggestions for use of these drugs and things to consider outside of a trial, because I know that a lot of people are thinking about this. So I think you need to understand your risk tolerance and understand that participation could prevent or delay participation in other clinical trials. And this depends on what that other trial's inclusion or exclusion criteria are. So let's say somebody were to try on their own with the advice of their clinician, memantine, but then a trial comes along with radiprudil. You may be excluded from participating in that trial because you've already been exposed to one of these drugs. And of course, knowing the functional status is imperative. And you need to work with your clinical team to determine ahead of time, what are you going to use to assess safety and efficacy? And it's important to understand that some of these things may or may not be covered by your insurance. Come up with ideas for safety studies, like what sort of blood work needs to be done. We need to follow heart studies like EKGs, need to follow EEGs. What sort of studies to measure neurodevelopmental efficacy need to be followed? Vineland's, Bailey's, Mullen's, just pick something. And then how often do these things need to be measured? My thought is if you're going to start a drug, you need to have an exit strategy. And that means, okay, when are we going to decide at what point, okay, it's not working, or it is working? If it's not working, we need to know how to quit. You don't want to just keep adding medicines that aren't of clear benefit. And of course, please have your clinical team contact us for any suggestions. And I think the registry is built such that you can enter, when you enter your follow-up information, if you've been on one of these drugs, you can add that information to the registry. So at least there might be some mechanism for us to be able to track these things going forward. So that's what I have. I really am hugely thankful to all of you families. I know that filling out those questionnaires online can be painful and can feel like you've lost valuable time, but I think that they're going to be really important for moving things forward with understanding a lot more about gain of function, loss of function, natural history, and even potential therapies. Thanks hugely to my partners, Steve, Dr. Park here in Colorado, Jennifer Bain at Columbia, Johannes Lemke, and Dennis Lau. And of course, our funders, past, present, and over the years. So thanks very much, everybody, for your attention. And Steve and I look forward to your questions.
Yeah, with that, we'll start questions. I have one in the chat, so if people want to keep populating the chat with questions, that's fine. If somebody wants to, unmute and voice, that's fine. But I thought I'd start with the one that is in the chat, and that is wanted to ask about number one, inhibitors. Is there only radiprudil, or are there also other companies that are looking for inhibitors? And then part two of that, the team in Barcelona completed a functional analysis and informed this person that the mutation is gain of function because the channel stays open too long. So in theory, radiprudil would work toward closing that channel. That's the question there.
Yeah, I can start, and Tim might have something to add. The class of drugs that radiprudil is in, there's many of those that have been made. It's a very vigorous class. We've known about it since 1988. Every major pharmaceutical company in the world at some point has been making GRIN2B, GLUN2B selective drugs. There are numerous ones in trial. You can go to clinicaltrial.gov and see who currently has compounds. So radiprotil is by no means the only 2B inhibitor. Now, most of those drugs are being made for other indications in adults. And so radiprotil is unique in the sense that the previous licensee of that compound, UCB Pharma, ran a clinical trial with 3 infants for infantile spasms in Paris some years ago. And UCB went through all of the necessary safety testing so that the European agencies, regulatory agencies, would approve the use in a pediatric population. And so radiprotil is a little bit unique. Among all the many hundreds of 2B inhibitors out there and the half a dozen that people are trying to look at, radiprotil, to my knowledge, is the only one that has a pediatric tox package associated with it, and therefore regulators would be willing to allow it to be tested in kids. But there are many, many other companies, and there's a Novartis trial running right now with a similar compound for depression. There's other companies out there, but that's the only one I know of being targeted for the population of patients in the GRIN2B community because of its pediatric qualifications. Tim, you want to add anything to that?
No, just that this has always been an active area of research. And I think that the other part of the question is that, and I think that this goes to Steve's talk, is that based on one functional assessment, it's been suggested that a particular variant may be gain of function. But as you saw from Steve's slide, is that it's important to apply the whole range of tests to understand, yes, it is either gain or loss of function. And it's anticipated that as the radiprotil moves towards a clinical trial in the US, is that the inclusion and exclusion criteria will probably use this latest, more complete classification in order to determine whether you're gain or loss and whether or not you could participate in such a trial. Is that a fair statement, Steve, based on from?
Yeah, I think so. And it may very well be the conclusion about the channel being open is the overriding property of that particular variant. But we have many cases where the channel is open, but the receptor doesn't, the machine's not built right and it doesn't traffic, it doesn't go to the cell surface, so that the ones that are at the cell surface work better, but 90% don't make it there. And then you end up with one of those conflicting situations we talked about, and you kind of have to weigh them off. Is the opening overweigh the lack of expression on the surface? And so you do have to look across, and maybe that data exists, from Chave and his guys. You just have to check in and see, have they covered these other bases, and is that a secure conclusion?
Did we answer that? I can't, I think you're muted.
Sorry. Yeah, I'm just kind of, I don't know. I'm just kind of reading it in the chat here. So what kind of medical team should we have? This is from Laura Chang. Moved a couple of years ago to a different state. Haven't been able to connect with any doctors other than establishing a new primary care physician. And what if the doctors we try to establish care with don't know anything about GRIN2B? Do we ask if they can connect with you to learn more about it?
So like with any rare disease, I think it's going to be hard to find, unless you land in a few places, doctors that are, have some experience with different GRIN disorders. So I think that absolutely, I mean, your medical team, you want folks that are okay with reaching out to others for questions, and that's what we're here for. I view that what we're doing here is a team sport, and I look forward to chatting with any clinicians. So.
Hey, this is Laura. I just wanted to follow up with that. Sorry, I don't have my video on, but when it comes to So should we establish a neurologist at the minimum or a geneticist? Like what type of doctor should we be connecting with?
Well, it kind of depends on the symptoms that your child has, but probably they need a neurologist. They may need a developmental pediatrician as well. But yeah, I think first of all, you need a good caring primary, and then to add to that, neurologist and maybe developmental pediatrician. I think if you already have your diagnosis, it may, and it depends on your geneticist. It's useful to connect with the geneticist at least once, but they may not be somebody that you necessarily need to keep going back to. It just kind of depends on, who's the most knowledgeable and helpful person that you think is part of your team.
Okay. I guess, sorry, just going back to so my daughter's she's almost 7 years old. She was diagnosed several years ago. We've already seen a geneticist and, neurologist. They say she has no seizures. We've already done EKG. We've done a lot of things already. And right now she's really stable. She has G2, but everything is good right now. And she just goes to ABA for autism. So that's why she hasn't seen anybody, which is why I'm asking at this point, if things have been quite stable, do we still need to establish care with a neurologist or geneticist?
I think so, because if you,
Okay.
Even if you just go see them once a year, they know you, and if something happens, then you're not struggling to, you've got an instant need, and then you're struggling to get a new patient appointment, and a lot of times the waiting time is 3 to 6 months depending on where you're at. But if they already know you, then you know them, you make the phone call, and you can get you have better access that way. So I think that it's useful to have, you see your neurologist on a yearly basis.
Okay. All right, that's helpful. Thank you so much.
Sure. There's a question. Would we just email Grin Research if we have a change? Yes. If you were in the registry, you should be getting yearly reminders to do your update. But if you enrolled a long period of, more than a couple of years ago, then you may not be part of the new version of the registry that sends you those reminders. So yeah, please reach back out to us at grinresearch@cuanchu.edu. And then there's a question. We would like to start our daughter on L-serine. Would you consult against starting L-serine under the supervision of our regular pediatrician? It's my opinion that if you're going to do this, it should probably be under your neurologist, because the main thing that I would be concerned about is making symptoms worse and potentially even causing seizures. And so I think that your neurologist should be involved. We've talked a lot of amongst ourselves. We would like to actually do a proper clinical trial with L-serine, but our basic hurdle there is coming up with the necessary funds to do a trial. Trials are expensive.
Okay, thank you. We actually just got, we have an appointment with Dr. Bain, but it's not till January, and we actually just found out that she can take us in October. So
Oh, great.
I'm assuming it's best to wait until then.
I would think so. Have a good discussion with Dr. Bain.
Okay, thank you.
Just curious, do you guys know Dr. Peter Crino? We just started seeing him after the seizure and more genetic testing done with him. Yeah, Dr. Crino's, he's name brand. You're in good hands.
Hey guys, I have a quick question. It's Lauren from GRIN. So about the clinical trials, when you were talking about, let's say we were in a trial hypothetically for meatine and radiprotol opened up and we might not be included, we might be excluded. So we tried the meatine a few months ago, my son Carter's gain of function, and it was terrible. It did not work for him. So we stopped it almost immediately. We weren't in a trial or anything like that. Our neurologist just got it for us and let us try it. Say that radiprotol did become an option like next year, would that exclude us even though we tried it like a year before?
So that really depends on the specific wording of, when the company writes this, I don't write it, your clinicians don't write it. The company says, this is the Bible for that particular trial. You've got to, when they write what they call a protocol for a trial, you have to follow it to the letter. And so they will write in what the inclusion and the exclusion criteria are. And I would imagine, that if you couldn't be on memantine and radiprudil in a trial, but they might say something like is okay if they were on it in the past, but they haven't been on it for at least 6 months. In other words, it's had plenty of time to come out of their system. So hopefully, I would imagine that a trial might look something like that, but I have no idea what sort of protocol somebody's going to come up with for a trial like that. It's probably going to have some sort of temporal, it's been such and such period of time.
Okay, that makes sense. Thank you.
But it certainly wouldn't be, there is going to be a washout period, probably months.
Okay, that makes sense. Thank you.
Hi, I guess I'll just talk because I can't figure out how to raise my hand. Thanks for answering my other question. I'm just curious from the scientific side, are there any animal models that you would consider using for functional, or is the difference too great between species? And are you looking at kind of some bioengineering approaches with, organs on a chip or something like that to kind of examine more functionalities? Because my son's variant really didn't show much as far as what you're testing right now. And but clearly there's an issue because whole genome, whole exome sequencing, there's no other mutation except for this one mutation that adds one stupid little carbon atom to the side chain of the amino acid, and yet he's severely affected. And so I'm just curious, what other types of model systems are you looking at to study other functions and loss of functions or gains of function?
Yeah, maybe I'll start on that one. That's a great question. It's a really great illustrative example of why we need to do more work. There are at least 15 mouse models of variants, and that may not sound like very many, but in the rare disease space, it's actually quite a lot. If we know about 400 variants and there's already 15 models of them, they're kind of broadly cross different properties. We have 6 of them at Emory, but they tend to be models that, for various reasons, have predictable effects. And so the field has not progressed, nor has funding, either from the government or elsewhere, have progressed to do the kind of work that you would ask. We make models where we know the variant has a really strong in vitro phenotype because we're trying to demonstrate that the animal, that there'll be a link to that, and we got to have something we can measure to show changes in the animal. So we've made very strong 2B gain of function, very strong 2B loss of function, truncation mutants that have really strong phenotype. And from federal funders, you need to do that. And so you can make hypotheses and argue that you're going to use the animal in a diagnostic purpose or to probe new molecular genetic solutions or new pharmacological solutions. It's very hard to get federal money at a basic research level to do exactly what you need done. And I don't diminish it at all. It's incredibly important. And there's a lot of variance in that category. But it's really hard to go off and get funding and say, we don't know what this variant does. If it does something bad, I want to make an animal and see what happens. And typically the government would say, well, that sounds really great, but we don't know that you're going to generate anything with our money. So maybe we'll give our money to someone we know will. And so there's a dearth of that kind of work. Now you mentioned organoids and iPS cells. There are a fair number of folks, moving in that direction, setting that up. GRIN's great at connecting people. GRIN2B is great at connecting folks. And so they've brought a lot of those individuals in, made them aware of what they can do with their systems. And that work is sort of percolating in the background. And I'm very biased on this, and Tim may have a different view, but my bias is the problem with that work that most people don't realize is how incredibly slow it is. It takes 7 weeks to grow an organoid to record from one cell, and then you got to stop, and that's 2 months. You can do 5 or 6 experiments in a year. And each experiment usually tells you, shoot, I didn't do this right. Maybe I need next time to do it another way. Well, now you gotta wait 2 weeks till you try that. So that organoid work is moving very, very slowly. And as it moves, the recipes, it's a dynamic growing field. The recipes for making organoids are changing. And so a year later, people aren't even making them the same way they were making them now. So maybe that field isn't quite mature enough to deliver precision, information that you would need. So that's a little bit about where things are. You should email me your variant and let me at least look at it and rethink about it for a little bit and get back to you about, maybe there's some other things that could be done outside those 6 assays that would be useful short of making an animal or iPSC cells.
Okay, great. Thank you so much for your thoughts.
Yeah, Tim, do you have anything to add to that?
No. So one of the things that Steve mentioned is that we don't know what's going on with the C-terminal variants, but we've thought a lot about how to try to address those, and it's something that's on our to-do list for actually this next year to see if we can penetrate that. But yeah, otherwise nothing else to add.
And just since I have you guys here, so I know that the GRIN2B seems to be really important for brain development. And there's people out there who think we can cure this. And I know that there's some, gene therapy techniques, and people are starting to think about things like that. But if the brain didn't develop properly in the first place, what are your thoughts on, even if you could replace a mutant gene, that it would actually cure it?
It's a great question, and it begs one to go ahead and show their hand on whether you're an optimist or a pessimist. And I'm an optimist. I don't think if the brain is miswired, you can go back and perfectly wire it. But the overriding feature, and I'm sure Tim will agree with this, of the brain as an organ is it exists to be a plastic signal machine and change. That's its whole, everything about it is to change. And that's why we learn and we adapt. And so my guess is that we will learn to improve things, and replacing certain defective parts may not set you back, to the way you would have been if it had never been made. But I think you might engage plastic mechanisms and things improve. And so I'm an optimist, a little bit of a realist. I don't think you can make it perfect, but I do think there's going to be an opportunity to make things better. I don't think, once you're miswired, that's it. There's no way to unscramble all the miswired circuits. I don't know, Tim, do you have anything you want to add to that?
Yeah, I think I'm always careful if we were to ever use such a word as cure. But I think all of us want things to be substantially better. But I think on the other hand, there are small inches with some of our children that if he could just do this, that would be really meaningful to me. And I think that that's just the other side of that. And I think that at the end of the day, it's gonna, we're gonna try whatever, there are gonna be small steps here and hopefully bigger steps with things such as gene therapy in the near future, but we'll see. But in terms of neurodevelopmental disorders and gene therapy, there are 2 open clinical trials in Rett syndrome. There are trials in Dravet syndrome and Angelman syndrome for things, another type of gene therapy called ASOs. That, I think that with regards to that whole idea of, development has already happened, what do things look like? I think we're gonna, a year from now, what we know is going to be very different with regard to that question.
Great, thanks. Yeah, I mean, because you never know how it will react, and certainly any small benefit you see immediately. It's always really nice to see some new things that, oh, I didn't know he could do that. So thank you for your thoughts.
We have a question in the chat. I'm just going to remind everybody it's about by my time 7:04. And so this will end at 15 minutes past the hour. So happy to keep asking questions until then. I just kind of want to be everybody to be aware of the time. So yeah, Laura's asking gentlemen if there's a neurologist in Texas at Texas Children's Hospital who you might know or recommend. Also wanted to know, how did you choose the 15 variants to be mouse models?
Texas Children's is a big group. There's a lot of really good people there. I don't know how you can go wrong. But I think the last person I talked to there was the head of the epilepsy service, and then also Gary Clark. He's the overall head of service there. So, but I think there's a lot of people there that are pretty familiar with GRIN disorders.
Yeah, I can comment on the variants. They were chosen by different labs for different reasons. When we first chose to make 4, we chose ones that were really strong gain of function, because what we were trying to demonstrate is that, in fact, these variants do cause clinical features of the patients, which in the early days was the pushback. In the very early days, when you would publish a paper, you have a variant, it would change function, and the reviewers and the community would say, well, how do you know that really underlied the child's seizures or the developmental delay? Yeah, you got a big effect in vitro, but I don't know if it's real. So the early choices, at least by us, were always strong changes that we could measure in vitro, and then we could go into the animal and show, actually, this is what's driving some of the differences we see in animal behavior that might be representative of the patient. So early on, that's how we selected some of them, and we selected representative gain and loss that way. And we have more recently just made an animal with a truncation to try to capture, like Tim said, more of the things that can go wrong in the C-terminal. And that's a truncation that isn't going to, the kind of things I do in the lab won't change. That truncation isn't going to change any of the 6 parameters I measure. It's going to change where it goes in the synapse, how it talks to second messenger systems and kicks off signaling to the nucleus. And so I think the animal selection is getting, past the obvious question, did the variant cause the problem? Which I know it seems hard to believe now that there was pushback on that, but there was early on. And now it's getting to grab larger collections of variants as representative, so that we can learn a little bit more about places where we're really thin on information.
And then I just another reminder about completing the survey link I put in the chat. If you're involved with GRIN2B Foundation, you haven't done that yet, we would love it if you could do that by tomorrow. It's really going to inform our foundation strategic plan moving forward. We want to make sure that we're serving our community the best way that we can. Other questions? So this, to answer, Chris, this is being recorded, and so we'll post this at a later time on social media, so you'll be able to access it that way. So a question there in the chat, is it a common misconception that it's quote unquote better to be a loss of function or gain of function? The symptom is very dramatically, is that correct?
It's what we want to quantify with your data from the registry. So I don't think we can answer it yet. Great question.
Any other questions? Well, I'd like to thank everybody for joining us this evening. Especially want to thank, and please do thank Dr. Traynelis and Dr. Benke for taking time out of their day to be with us and to present this information about gain and loss of function and why it's important for us to further the research for GRIN2B and frankly, multiple GRI disorders moving forward. So yeah, Tim and Steve, thanks a lot for joining us tonight. Anybody else have anything? A couple more things pop through, just some thank yous. Yeah.
Yeah, I'm just very happy that you did this. It's a wonderful presentation. Enjoyed it a lot.
I was just going to say, Phil, thank you for organizing this and all the work you do for the foundation, which is really exceptional. It's fantastic that you guys have pulled this organization, and it's a pleasure to be able to help even in a small way here.
Thank you very much. Leslie, I saw you jumped on. I don't know if you just came in or if you've been here and got kicked out and came back in, but we did record this entire, we're about done here. We did record the entire webinar, so we'll be sure to, we're gonna post this at a later date. So, all right, everybody. Well, again, thank you very much for joining us, and have a great rest of your week and a good weekend. Thanks a lot, everyone.
Bye.